Okay, good morning. I'd like to get started. Good morning, everyone. I'd like to get started. Thanks. Including my GSIs. Those of you who elected to stay at home and watch the webcast instead of coming here missed an opportunity to walk across the Cal campus on an absolutely lovely day. Okay, and those of you watching from some other exotic place. Is it too loud? Is it okay? It's good? All right, the sound. So today I'm just going to finish up a little bit about microscopy that we were talking about in my first lecture, and then I want to talk about introduce you to some of the sort of advanced microscopy techniques that are being used these days to make all the nice images that I showed you last time that appear in your textbook, for example. Then I'll give a sort of general introduction to the cytoskeleton, which is really the meat of what we will be studying and talking about for my lectures. And then we'll drill down a little more into actin filaments and talk about their structure and dynamic properties. So, advanced microscopy. So, I mentioned that the pace at which new discoveries are made in cell biology has, to a large extent, been dictated by the pace at which innovations are made in our ability to image cells and to see the details in the structure of cells. And so innovations are being made that increase our sensitivity, our resolving power. Okay, so some of the things that increase our sensitivity are the development of objective lenses with a higher numerical aperture, development of faster, more sensitive cameras. Everything is digital these days, so we use CCD cameras, like in your digital cameras that you probably use for your own snapshots and whatnot. So higher sensitivity CCD cameras, and then that's sort of on the instrumentation side. And then there also are brighter and more stable fluorescent probes for fluorescent microscopy. And for the most part, when we look at cells these days, we're using fluorescence because it's a technique that allows us to tag specific molecules and to watch those specific molecules. Now, having better sensitivity is important because that allows us to detect more molecules that are lower in abundance in the cells, but also it allows us to turn down the intensity of the excitation light that excites the fluorophores so they can fluoresce. And that's important because we want to, if we want to image living cells, we want to protect them from photo damage. And so we want to use the lowest level of light possible and still be able to detect specific molecules. And finally, higher sensitivity is also important because it allows us to see things with greater time resolution. If we're looking at processes in living cells that are occurring very quickly, things are moving around, chromosomes are getting separated, vesicles are budding off from membranes, molecules are entering the nucleus through nuclear pores. These are happening on often timescales of seconds or even fractions of seconds. And the higher the sensitivity of our imaging system, the more frames we can capture per given interval of time. And so we have better time resolution, okay? Not only spatial resolution, which I'll get at with resolving power, but also time. Okay, so let's see. Actually, resolving power. I made one mistake. No one caught me. You should have. I meant to put the higher numerical aperture under the resolving power. Okay. So remember, the higher the numerical aperture, the greater the resolving power of our microscopes. In addition, I'll talk about in a moment, some new types of microscopy that are also improving our resolving power. Other kinds of innovations include automation. So, for example, there are robotics that prepare samples and actually collect images. There are automatic focus stages. And then there are automatic image acquisition systems. And this has allowed several new types of endeavors to take place. For example, drug companies now use cell-based screens where they add chemicals to living cells, and they actually use imaging to screen for molecules that cause very specific effects on those cells. Diagnostic companies also use automated imaging to look at samples from biopsies, for example, in an automated fashion to look for signs from the structures of the cells of certain diseased states. Okay, and so then I mentioned that there are a variety of new imaging approaches that are used. Approaches or techniques. And so these are things like confocal microscopy, which is not so new anymore. There's things called palm microscopy, and there's structured illumination. Which you may be hearing about in your reading. So palm and structured illumination are very new techniques that are helping to really push the resolution limit of light microscopy by using both a combination of new types of instrumentation and also new computational methods to analyze the outputs from these imaging approaches. I'll just say a word about confocal microscopy. You know, often you're trying to look at a structure inside some three-dimensional object. Say it's a large egg from an amphibian or a developing embryo, and you want to see some feature down inside of that large three-dimensional object. Well, one of the problems is that when you have a cone of light and you bathe that object with this cone of light, you're illuminating the area both above and below the detail that you want to see. And so while you're trying to focus on this part, your ability to see that part of your sample is impaired because there's out of focus light coming from above and below that feature that you're looking at. And so the way confocal microscopy works is it uses a laser to focus the light in three-dimensional space on a particular point, the point of interest, and at the same time, the objective that gathers the light gets focused at that same point, so it collects light from that same point in space, eliminating this problem of all of this light above and below your sample. Okay? So confocal microscopy is a very powerful technique for being able to discern details in a three-dimensional image. Okay? And you can actually set up the confocal microscope to, section through and sample the whole three-dimensional object, but doing it one point at a time, and then using a computer to recreate the whole object. Okay? So these are really powerful techniques. And going hand in hand with sort of the instrumentation and chemical developments that are pushing microscopy forward are also some computational advances. Okay, and that's what happens when you gather the light. So these days, microscopes are almost all use digital imaging, just like with your digital cameras, and just like probably most of you have manipulated your images using Photoshop or iPhoto or something like that to increase contrast and accentuate other features. You can do similar things and even more sophisticated things using software or algorithms that have been developed for analyzing microscopy. So one of the things they can do, so they can increase contrast, they can correct for optical aberrations. So at some point, they've really come very close to perfecting the art of making lenses for microscopy. But every lens at some level is imperfect. It has some kind of aberration. And so what you can do is for a given lens, a given piece of glass, is you can sort of teach the computer what the aberration is on that particular piece of glass, and the computer can compensate for that aberration and can make the image sharper. There's something called deblurring or deconvolution. And this is a computational approach to doing what I told you the confocal microscope does. Basically, the computer algorithms can understand where if you're looking at an image that's blurry, it can sort of calculate back what a sharper image would have created that blurred image. And completely by the magic of computer software, it can actually sharpen your image to show, to eliminate the blurring of this sort of out-of-focus light. There are a number of techniques for automatic feature recognition. Like I knew of a drug company that was looking for molecules that blocked in mitosis. And so they had some software engineers write software that allowed them to, in an automated manner, screen through images of cells and find cells that were blocked in mitosis. And so then they could use small molecule inhibitors. They were trying to find a cancer drug that blocked cells in mitosis because cancer cells are rapidly dividing. So they're using feature recognition. A lot of this stuff actually comes, some of it from astronomy, where astronomers have developed software, and also from the defense industry. These are guys who have figured out ways from satellites to see something like the size of a deck of cards, but even more impressive, they use feature recognition to be able to figure out, if there's a tank underneath a tree or something like that. And so using those same kinds of computer algorithms and turning it towards biological specimens, you can actually find things that are important for learning about cell biology and for medical diagnostics. There's also tracking of dynamic structures. So if you make a movie of, say, vesicular traffic, a computer can latch on to, say, a given vesicle, and it can track that vesicle through its life and follow it. And then it can give you all sorts of quantitative information about the life of the vesicle and where it was when it first appeared and where it was when it disappeared and so on and so forth. And then another thing that you can do is to make three-dimensional reconstructions. Okay, and I showed you in that slideshow last time that using computers you can take three-dimensional data sets. So you have some big complex structure, and it's a three-dimensional structure, and then you can either get data by collecting sort of either optical sections or actual physical sections. Like in EM, they'll cut sections through a thick specimen like a cell or even a whole tissue. And then you can collect all that data, and the computer will reassemble it for you. Okay? There's two kinds of basic approaches. There's sectioning, which can be either optical. In other words, you're focusing on different planes at a time and then having the computer assemble those all. Or there's actual physical sectioning. Okay? Or in contrast, there's something called tomography, which refers to a process in which the sample is tipped from side to side, and the sample is and the images are collected always from the same direction, but the sample is rotated so you see it from different vantage points. In either case, the computer can then take all that information and reassemble it into the kind of three-dimensional structure I showed you last time. Okay? And that's really important. You know, a lot of times when you look at, say, an electron micrograph in your textbook, it can be really deceptive. So suppose you had an object like this, and you used electron microscopy to take one section through it. Okay? And this could be, in real life, this could be something like a mitochondrion. You know, what would you see? You would see, when you took this section and then turned it on its side, you'd see, you know, something here, something a little bit bigger here, and something a little bit smaller here, you know, when you look from the top. And so you might conclude that there were 3 different mitochondria here that were all kind of small in size. But in fact, what's often the case, especially with membrane organelles, is that you get the impression that there are a number of small organelles with mitochondria or the endoplasmic reticulum, but actually these things are all connected together by some kind of branching network. And you can only really get a sense of that if you can image this thing in 3 dimensions. Okay? So that's all I wanted to say about microscopy. Are there any questions on any of that? Yes? So PALM, I didn't go into PALM, but PALM and the structured illumination are 2 techniques that are pushing the resolution of light microscopy. Okay? They're very sophisticated techniques, and they work in very different ways. And I'd be happy to talk to you sort of outside of class about it, but it would probably take a good part of a lecture to explain either of them. But basically, it's getting beyond that 200 nanometer limit intrinsic to light microscopy and pushing it down and down, you know, down to now like 15 nanometers by using these approaches. Okay? Other questions? Okay, let's switch to talking about the cytoskeleton. Okay, so what is the cytoskeleton? The cytoskeleton is a network of protein polymers that are found in the cytoplasm of cells. Okay, and this includes actin filaments, which we'll talk about first in the class. It includes microtubules, includes intermediate filaments, and then a 4th class that often doesn't get discussed, the least famous class, but we'll just briefly mention it, is called septins. Okay? So these are all different systems of protein polymers that serve specialized functions that are very important for the biology of cells. For a long time, it was assumed that, based on a lot of evidence, that the cytoskeleton was a eukaryotic invention, okay? Because people had looked for but failed to find evidence for any of these proteins in prokaryotes, in bacteria. However, over the last decade, it has become clear that the cytoskeleton is actually a very ancient invention, and that prokaryotes have, at least the first two filament systems, they have proteins related to actin and microtubules. So related to actin in bacteria are proteins called MREB and PAR-M. And related to tubulin is a protein called FTSZ. Tubulin is the subunit protein that makes microtubules. Now we'll be learning about we're going to focus, this class focuses on eukaryotic cells, but just at the beginning I wanted to talk a little bit about origins. One of the interesting things, so MREB functions in the sort of cell shape of bacteria, but PAR-M functions in chromosome segregation. And that's kind of interesting and surprising because most of you, I think, probably know at this stage that tubulin and microtubules in eukaryotes are involved in chromosome segregation. So what does FTSZ do? Well, FTSZ is involved in cytokinesis in bacteria. Okay, which is the opposite of what's seen in eukaryotes because actin mediates cytokinesis, not tubulin. So it would appear then that in general these proteins have evolved to make polymers, and that over time through evolution cells have found different uses in different contexts for these systems of polymers. Okay, so then what are the functions of the cytoskeleton? And now, henceforth, we shall talk about the cytoskeleton in eukaryotes. So what are the functions of the cytoskeleton? Well, one function is in cell shape. So, for example, you could have a neuroblast cell, which is a precursor to a neuron, or nerve cell, or a platelet, which circulates in your blood and has a very nondescript kind of shape. And then the neuroblast can differentiate to get a very pronounced elongated shape, the specific nature of which is critical for establishing the wiring of the nervous system. So this is a nerve cell, and it has an axon and a dendrite, or more multiples of these. And this platelet circulates around for ever and ever in your blood, maintaining this shape until, in response to a wound, it is activated, and then in an amazingly fast short amount of time, it adopts a very irregular shape as part of the wound healing response to block up a wound. In both cases, these dramatic changes in the shape of the cell are completely driven by changes in the organization of the underlying cytoskeleton, or the regulated assembly of cytoskeletal filaments. Okay? In this case, in an almost explosive manner, the actin cytoskeleton reorganizes to change the shape of the cell. And it's an emergency situation. There's a wound. These cells become adhesive, and they change their shape to fill up space. This cell adopts a specific shape to establish the wiring of the nervous system. In both cases, the underlying important question is, how did the cytoskeleton do this? What properties of the cytoskeleton allowed it to drive these changes in the shapes of these cells? So a second major function of the cytoskeleton is in controlling the internal organization of cells. Remember, I told you last time that a cell is not simply a bag of enzymes, that it consists of different microenvironments, and those different microenvironments within the cell have distinct places where they should be in the cell in order for the cell to function properly. So the second function is in setting the internal organization of cells. So again, if we look at this nerve cell with its axon and dendrite, these different regions of the cell have different biochemical compositions, okay? And that has really important functional consequences. The axon is presynaptic. It's making, it's sending out neurotransmitter to the next cell, and the dendrite is receiving neurotransmitter. Okay, so in one case, you have this system that's sort of jacked up for secreting neurotransmitter. In the other part of the same cell, there's a region that has receptors for the neurotransmitter. So how is it that within one cell you can have different regions that are specialized in terms of their function? The way you can have different regions that are specialized for different functions is that they have different biochemical compositions. And the way you have different regions of cells that have different biochemical compositions is by attaching to the underlying cytoskeleton molecules that are asymmetric. Another example of that would be in an epithelium. So here you have a sheet of epithelial cells. And so on and so forth. They're connected to each other by tight junctions, so they make an impermeable barrier. On one side, you have the gut. On the other side, you have what's essentially the blood. And so nutrients would be taken up here, and waste would be sent out. So this cell is very asymmetrical again because different faces of the cell, the so-called apical and basolateral, have different functions, and the way they're able to have different functions is that they have regional differences in their molecular composition. And those regional differences allow them to transport nutrients in one direction across the layer from the gut into the blood, and to get rid of the waste in the other direction. Okay? And the cytoskeleton is absolutely crucial then for this internal organization. One other example of internal organization is if you just look at a generic cell, you're used to seeing the nucleus. Nuclei are usually blue, right? And some cytoskeleton, and then there's various organelles like the Golgi apparatus and the ER and whatnot. And so how do these different components of the cell have this kind of organization? Well, if you dissolve the cytoskeleton using drugs that bind up cytoskeletal proteins, all of the organization gets lost almost instantly. For example, if you depolymerize the microtubules, the Golgi fragments into little vesicles. Okay, it's no longer held next to the nucleus. So again, the cytoskeleton is dictating the internal organization of the cell. The final general functional class of the for the cytoskeleton, or role for the cytoskeleton, is in force production. So the cytoskeleton is important for motility of cells, and we'll learn a lot more about this. Actin is responsible for the crawling movement of cells of animal fibroblasts or amoeba. Microtubules are important for swimming motions of flagella and cilia. The force production is important for mitosis, for separating chromosomes, for cytokinesis, and then for transport of organelles, moving organelles around within cells, so organelle transport. So to understand these three features of cells, we have to understand the underlying cytoskeleton. So for the cytoskeleton to have these functions, it has to have sort of dual properties, and they seem kind of contradictory. So in a general sense, the cytoskeleton first must be rigid. Only by being rigid can it resist compressive forces to hold out the cell in these shapes. Okay, the membrane wants to be, wants to form a spherical structure. That's the energetically favorable state. There's a big energy cost to push the membrane out and to hold it out. In order to do that, the cytoskeleton must be rigid, must be able to resist compressive forces. The second thing, though, is that the cytoskeleton has to be dynamic and intrinsically unstable. And that seems paradoxical. Okay? Because cells are not static. They're changing their shapes constantly. This platelet, within a matter of seconds, changed from a spherical shape to this elongated shape. But a fibroblast cell crawling across a substrate, maybe it's following a chemical gradient, will follow it in one direction, but if it encounters a stronger chemical gradient of a chemoattractant coming from another direction, it will immediately change directions. And the whole time, the cytoskeleton is assembling and pushing the cell in one direction, but then it has to, like that, change directions and assemble its cytoskeleton in another direction. So how is it that the cytoskeleton can be both rigid and dynamic and unstable? That's going to be one of our key questions. So let's just sort of enumerate some of the questions as we move through specific cytoskeletal filaments that we're going to ask about all of these filaments. You could consider this list of questions sort of a good study guide for what I'd like you to learn. So these are sort of general questions about the cytoskeleton. So first, what are the functions of the different cytoskeletal filaments? Actin, microtubules, intermediate filaments, septins. Second, what are the intrinsic properties? Okay, that allow each cytoskeletal system to carry out the functions it carries out. How does each different cytoskeletal system assemble and disassemble? And then, what are the roles for interacting proteins? So for example, actin filaments are made of actin, and microtubules are made of tubulin, but they interact, each of these filaments interact with lots of different proteins. What do those proteins do? What are they for? And then the fifth question is motor proteins. And what we want to know is what are their functions and how is it that they convert chemical energy into forces? Functions and force mechanism. And we'll be mainly addressing the force mechanism for myosin. Okay, so those are the main questions that we want to address. Now, as we continue to sort of drill down, we're going to talk about our first cytoskeletal system, about actin filaments. Okay? Their structures, their dynamic properties, and their functions. Actin. Okay. Like all of the filaments that we're going to talk about, these are polymers of a single kind of protein. And so actin can exist in a monomeric form, and it can exist in a polymeric form. Okay? The monomer is often referred to as G-actin, and G stands for globular. It's a globular form of actin. And the filament is often called F-actin, and that means filamentous actin. Now, the filament is actually not a single strand like this, but it's as though there were two strings of beads that were sort of twisted together. For simplicity, I'll often just draw them in this manner, but they're actually kind of a twisting pair of filaments that make up this filament. Okay, so the subunit of actin, the actin protein, is a 42 kilodalton protein. When it assembles into filaments, these filaments have a polarity. They have a physical polarity, which also gives them a functional polarity. They have one end, which is often referred to as a plus end, and the other end, which is referred to as the minus end. Why do they have a polarity? Well, let's look at the actin molecule itself. The actin molecule itself is asymmetric. A really interesting and important feature of actin is that it binds an ATP. And an absolutely critical question to understanding how actin performs its function is to understand why this protein that forms polymers should bind to ATP. And what we'll see is that when it binds to ATP in polymers, the ATP gets hydrolyzed to ADP. And I will elaborate much more on the consequences of that ATP hydrolysis in the next lecture, but you can think of it as constituting a chemical switch for the actin, because ATP actin likes to assemble, but ADP actin does not. And in that one key fact comes the clue to one of these questions I asked, which is, how is the, disappeared somewhere, what is responsible for this duality of filament proteins where they can make stable structures, yet they can also be dynamic and unstable? And the idea is that the ATP acts as a switch, and we're going to spend more time on that, okay, in the next lecture. But let's come back to polarity, the polarity of this filament. This actin molecule is asymmetric, and when it assembles into filaments, it assembles with the same orientation throughout the filament. And so that means if you look at the filament from one end versus from the other end, it's different. Okay, the two ends are different. So we call one end the plus end and one end the minus end. This filament is 7 nanometers thick. Okay. Suppose we took this actin and we labeled it with a fluorescent dye and then allowed the actin to assemble into a filament that's 7 nanometers thick. Would we be able to see this filament using a fluorescence microscope if it's 7 nanometers thick? You see some people shaking their head no. How many people in the class say no, you cannot see this filament? Raise your hand. Okay. How many people say yes, you can see this filament by fluorescence microscopy? And then how many of you either aren't paying attention or don't know the answer, don't want to make a guess? Well, there's a fourth class. I don't know what they are. They're just asleep. But okay. But it seems pretty much split, half and half, that we can't see the filament or we can see the filament. Somebody who says we can't see the filament, tell me why you think that. Come on, be brave. We're all friends here, right? Yeah. You'd see a string. But you would see something, you're saying? Okay. Yeah. Okay. So you'll see something, but you won't be able to tell what it is. So I think you guys are all really actually have the right answer. The molecules are fluorescent. And so you're going to see a line of fluorescence. You're going to see a line that is glowing. In this case, it's red. But what is the limit of our resolution? 200 nanometers, and this is 7 nanometers. Would we be able to tell, say there was a second actin filament right next to it. So now you have 14 nanometers. Would you be able to tell the difference between this and this? No. Okay. So what you're going to see a blurry fluorescent line. You're going to see a fat fluorescent line that's maybe like 200 nanometers thick. Okay. You're not going to be able to tell where those photons are coming from, but you're going to see a line of photons. Okay? Is that clear to everybody? You have the sensitivity to detect the actin is there, but you don't have the resolution to see its size or to distinguish whether it's one actin filament or 2 or 3 or 4 even. Okay? Yeah. Either. So when I say one actin filament, I do mean the 2. I just can't draw 2 all the time. That is 7 nanometers is the 2 twisted together, right? Yeah. Okay? All right. So, good. Now, how do we know that these filaments are polar? Well, we can look at them with the EM. In the EM, you can get down to like 2 nanometers resolution, but it's really hard to see this intrinsic polarity of the filament. And so there's you actually can see it with really good fancy EM, but there's a trick that makes it much easier to see the polarity. And that's because, I'm going to draw the actin as really simple again. Because myosin, we're going to study that on Friday, myosin binds to actin. Okay, it's a force-producing enzyme, and you can use a protease to cut off a piece of myosin, the part that binds to actin. And so this piece is called myosin S1. Okay, just for historical reasons, that's its name. And you can take this piece, this is the actin binding sort of business end of myosin, and you can bind it to actin filaments in a test tube. And when you do that, the myosins bind at an angle. And that angle accentuates the intrinsic polarity of the actin filament such that these guys sort of open up at the plus end, and the pointed end, well, the minus end, looks pointed. And from that, you get a second nomenclature. The minus end is often called the pointed end, and the plus end is often called the barbed end. Okay, it's like the barb of an arrow, bow, from archery or something. Okay, so that's how you can see that intrinsic polarity. Now, I told you that polarity has a functional consequence. Okay, if you're an actin monomer looking to join this filament, and you look from this end versus this end, the two ends look different. Okay, and that has a functional consequence because actin monomers prefer to bind to this end of the actin filament. And so if you take a filament like this and you make a short filament and say you decorate it with myosin, okay? You prepare something like this, and then you flood in a bunch more actin monomers, okay? This is an experiment now. And you ask what happens. What happens is that the actin will assemble 10 times as fast on the plus end as it will on the minus end. Okay? So the plus end is the preferred end for adding actin monomers. Okay? So when we study actin, as we get deeper into actin, we need tools to study actin assembly. One of the important tools at our disposal are inhibitors or drugs that perturb this assembly reaction. Okay? And we commonly talk about 3 different drugs. We talk about cytochalasin. We talk about latrunculin. And we talk about phalloidin. These are all natural products that come from sponges and mushrooms and things like that. Each of these drugs has a different mechanism of action. Cytochalasin is really interesting because cytochalasin caps the barbed end of the actin. Okay? So basically, it binds and blocks assembly from occurring at the preferred assembly end of the actin filament. So it will attenuate actin assembly. So it's a barbed end cap. Latrunculin binds specifically to the actin monomers, okay? And it prevents those monomers from assembling. It basically takes them out of the game, okay? So this is referred to as a monomer sequestering protein, or drug. Okay, and then finally, phalloidin is this mushroom toxin that binds to actin filaments and it stabilizes the filaments. One of the important properties of actin filaments is that they can assemble, but then they can rapidly disassemble. Phalloidin binds to the filament and prevents disassembly. One can attach a fluorescent chemical to phalloidin, and it's one of the favorite ways for visualizing the actin cytoskeleton. So, for example, you can use a rhodamine-labeled phalloidin, which is red, and you can bind that phalloidin to actin filaments, say in a cell, and the actin filaments will all glow red. And I showed a very striking micrograph last time of a cell whose actin filaments had been labeled with rhodamine phalloidin. Okay, I'll break here, and we'll continue talking about actin next time.